Dehydration and volume depletion. Plasma osmolality is about 290 milliosmoles per liter, contributed mainly by sodium, 140 millimolar per liter, and its accompanying anions. In dehydration, water is lost from the body. The rise in osmolality that occurs in the plasma, also sodium rises, causes water to initially move out of the cells along the osmotic gradient. Thus, cell volume is initially reduced, but cell homeostatic processes subsequently return it towards normal by taking up solute. In dehydration, water is removed from the plasma, and thus hematocrit and albumin, which have not been lost, have a higher concentration. In volume depletion, water and electrolytes are both lost, and thus there will be little effect on either sodium concentration or osmolality. As osmolality is not altered, there will be no force to pull water out of the cells, and cell volume is not affected. In volume depletion due to blood loss, the hematocrit acutely is the same, but the resultant fall in blood pressure causes fluid to come out of the interstitium into the vascular component, and albumin and hematocrit both decrease. When there is volume depletion due to electrolyte and water loss by vomiting or diarrhea, there will be little or no effect on plasma osmolality or sodium concentration. However, there will be a small increase in hematocrit and plasma albumin because the volume is lost from the extracellular space, and as blood cells and albumin are not lost, this increases the concentration. In volume depletion, forces are activated that retain sodium and water in the body. The sodium retention works to a major extent by the renin-angiotensin-aldosterone system, which is activated by a fall in blood pressure caused by volume depletion. In dehydration, the high osmolality activates ADH secretion, which causes water retention. As there is also volume depletion, this activates the renin-angiotensin-aldosterone system, which causes sodium to be retained. This retention would tend to cause a rise in sodium concentration, which is already high, but the water retention would correct this. There is no effective receptor that monitors and controls sodium concentration by altering sodium excretion. Sodium-retaining hormones are predominantly regulated by the volume and blood pressure. Initially, in blood loss, the hematocrit is not altered, but falls as fluid comes in from the interstitial space. Water balance. Vasopressin, also called antidiuretic hormone, is the principal compound controlling water balance by decreasing water output by the kidney, and thus decreased urination. It perceives the need by monitoring plasma osmolality, and if this is high, vasopressin is secreted. Vasopressin is formed in the hypothalamus and travels down axons to the pituitary, where it is stored. Plasma osmolality is the usual factor regulating vasopressin release, but other factors alter the release. Pain and emotion release vasopressin together with the other posterior pituitary hormone, oxytocin. Alcohol inhibits the release of vasopressin and thus causes diuresis. A low plasma volume also releases vasopressin, which in high concentration can cause vasoconstriction. These different factors can overcome the usual physiological control of osmolality. Osmoreceptors in the hypothalamus monitor the plasma osmolality and send a signal down the axon that releases vasopressin from the posterior pituitary gland. Vasopressin travels by the blood to the kidney and binds to a receptor on the basolateral membrane. By a series of cellular events, alters the permeability of the luminal membrane to water, thereby increasing the water permeability of the collecting duct, and due to osmotic gradients created in the kidney, causes water to be retained by the body, which is antidiuresis. This provides the other name for vasopressin of antidiuretic hormone. Vasopressin released by the pituitary gland binds to a receptor on the basolateral membrane and activates adenyl cyclase, which increases cyclic AMP levels in the kidney. Thus, by a series of reactions, some of which involve calcium, causes microfilaments to contract and insert pre-formed water channels, aquaporins, into the luminal membrane, thus increasing water permeability. A high plasma osmolality is the important physiological stimulus causing vasopressin release. Urea in plasma in a normal person only has a concentration of 6 millimolar per liter, and thus contributes to only a small part of plasma osmolality. Even if plasma urea is elevated to 30 millimolar per liter, it would not have a significant effect on vasopressin release, as membranes, including those of the osmoreceptor cells, are permeable to urea. If there is excessive ADH, water is retained and the osmolality and sodium concentration would fall, hyponatremia. If there is no ADH, water is lost, and osmolality and sodium concentration would rise, hypernatremia. While ADH is released, if the plasma volume falls, the most important factor to restore volume is retention of sodium by the renin, angiotensin, aldosterone, and other salt-retaining systems.